All right, good afternoon. So what I'm gonna do today is more a broad overview of kidney development. Okay, so it's more focusing on the events that happen. So that's about the molecular player. So when we talk about kidney development, so we and most of us are working on actually one of the kidneys. In fact, during development, there are three kidneys that formed sequentially. So they are pronephros, mesonephros, and metanephros. So today we'll mostly focus on the metanephros, which we also call it metanephric kidneys or adult kidney, the permanent kidneys. But so I will just give a few brief words on these two kidneys, the pronephros and the mesonephros. So in mammals, pronephros and mesonephros are both vestigial organs. So they don't function postnatally. They do not even contribute to any parts of the adult kidney tissue. But in other organisms, for example, fish, zebrafish, that's one of the model systems people also use to study kidney development. Pronephros is the kidney, that's a functioning kidney. And then for mesonephros, for example, like so frogs, like xenopus, people use that as also the model to study kidney development. So their pronephros and mesonephros are functioning organs. So because of that, I'm just gonna say very briefly in mammalian system, and for this, the time, as you can see, whenever you see the embryonic, this is mouse time point. Okay, so all the kidneys actually, the three kidneys, pronephros, mesonephros, and the metanephros, they all derive from this mesoderm called intermediate mesoderm. And then so at around E8.5, that's the time when you start to see pronephral formation. And so you will first have this cord that's called nephric duct. It forms from a nephric cord and then epithelize later. And then so this forms the nephric duct, and then you will get pronephros. Okay, so pronephros in mammals, so it just fully developed. In mouse, it basically you just see some condensates, like about 3 condensates never fully develop. But in humans, you actually do see pronephric tubules. And the thing is, so it starts probably around 4th weeks of time, you first start to see pronephros. And then it probably lasts till 4 months-ish in the fetus. So the thing is, when it develops from rostral to caudal in this direction. And then while the caudal ones form, the rostral ones already start to degrade. So anytime you probably only see 6 pronephros, but eventually they are all gone, so all degenerates. So later on, so in mouse, around E9.5, you will also start to see mesonephros. And then for the mesonephros, in mouse, we will see about, in total, 18 mesonephric tubules. And the interesting thing is that there's 6, the rostral 6, 4 to 6, roughly, is connected to the nephric duct, just like when you see the kidneys where all the nephrons are all connected. But the rest of 12-ish, they are like just hanging there. They don't get connected to the nephric duct. So these ones actually do have some functions. They actually do, like filter urine during embryonic life. But eventually by E15.5, most of this are already degenerate. You do have leftover of mesonephric tubules, and those actually do not contribute, as we just earlier mentioned. they do not contribute to the kidney tissue, but they actually contribute to the reproductive system. So in males, they contribute to epididymis and the perididymis, and in females, they contribute to epiophyra. All right, so, and then, so that's that's all I'm gonna talk about for these two early kidneys, and then we'll focus on the metanephric kidney. And then all that starts is in the caudalmost part, along this intermediate mesoderm and nephric duct. So this part is called metanephric mesenchyme. This is the condensed mesenchyme. One can actually see it just by looking at it. And then in humans, that development starts at around the 5th week of gestation. And by 32 to 36 week of gestation, it completely finished. Okay, so for humans, there is no postnatal new nephron formation. So that's different than mice. So in mice, it starts at around embryonic day 10.5, so E10.5, and then complete development by P4, meaning after that, you don't have new nephron being induced. So what you have by now, that's the maximum number of nephrons are formed. But that doesn't say by P4, so basically we will come back, when we go further into the presentation, it will more make sense what I said of P4, so it's actually nephron injection, but that's not the entire nephron, maturation and differentiation. So, but what one can say is for human postnatally, it was a term maybe, and for mouse, after P4, you don't have any more nephron formation. So one use the word nephron endowment to show like that is your the developmentally the maximum number of nephrons formed. So individually it's all different in number. We'll come back to that later. So the early events. The first event that really marks the onset of metanephric development is this invasion of from the nephric duct and the nephric duct into this metanephric mesenchyme. That's called ureteric bud outgrowth. So this marks the onset of development. And then after this, it's gonna be a repetitive events of two processes which actually involves the molecular interaction between the ureteric bud epithelium and the metanephric mesenchyme later on. It's called cap mesenchyme. So their interactions drive that event, and then in which the forms at least defines the cortex of the metanephric kidney. So since we talked about that the first event in the metanephric kidney development is the ureteric bud outgrowth. So this so first talk about this process. This UB outgrowth is actually a pretty vulnerable event. And then so that in humans, actually you can get, and then in mouse also, even sometimes spontaneous, you can get unilateral or bilateral renal agenesis. So shown here is actually a human, it's a radiograph of a person. So there's only one kidney. So you have unilateral renal agenesis. It's actually a silent event. So the person with one kidney, you can walk around healthy, generally speaking. So there's some compensation. So unilateral agenesis happens about 1 in 1,000. And that's actually in asymptomatic palsy. So the meaning like you don't, you go there even without problem, you can find with this kinds of occurrence. So it's a pretty high event. If it's bilateral renal agenesis, the embryos can, so the embryo, the fetus can still develop healthy. So we don't need kidneys for our embryonic and fetal life. So it's not necessary, but there is a consequence So the amnion fluid is actually the urine of our own, the fetus urine. So we are, like swimming in that. And therefore, if you have a renal defect, that can lead to oligohydramnios or anhydramnios. And then you have the Potter syndrome kind of the facial feature. So you can develop, but with some consequences. But postnatally, clearly, unilateral is fine, but bilateral is not compatible with life, right? And then so when we do mouse work, for example, you can tell if an animal die within 24 to 48 hours after birth. That's one sign of actually kidneys are not so both of the kidneys, so there's almost pretty much no renal functions. So we talked about, if you don't have UV outgrowth. So what if you have too much? Okay, so that's not better at all. So if you have, so if one develop with, multiple UV outgrowth, that can lead to duplex kidney, double ureter, and ectopic ureter. And then the problem with that is one can have horseshoe kidneys, so like with two, and then worse than that. So this kidney, in fact, at least histologically, doesn't look as bad. A lot of times one ureter and kidney is associated with hydronephros and hydroureter because the position of this UV outgrowth, then this UV eventually is not going to be connected properly with the bladder. So then you have urine buildup. So, okay, so then we go down to the subsequent event from UV outgrowth. Once UV invades into the metanephric mesenchyme, then two reiterative events occur throughout the rest of kidney development. And that is UV branching morphogenesis and nephrogenesis. So we'll first talk about UV branching. So for UV branching, its purpose, so what it achieves, first of all, through this branching morphogenesis, it establishes the entire collecting duct system. And then, so this branching, so the kidney branching is almost always bifurcation. And it always happens at the tip of the UV. And that's different than the lung. So the lung has lateral branching. So it is from the stalk part, you get branching morphogenesis. And then, so this is a process that is, so you go in and then you bifurcate. Initially, this is like in a mouse, you look at it, this is perfect. So it's a typical structure you will see at embryonic day 11.5. If you dissect, you always see that. And then it further bifurcates like this, and then you have this ampulla that's a more swollen kind of feature. That's the tip. This entire branching morphogenesis is induced by the cap mesenchyme. So the cap mesenchyme surrounding it here, it secretes signals that interact with the receptors that's expressed at the tip, and that induces branching morphogenesis. So, okay, let's see next. I want to show you a movie. Does it work? Doesn't seem to. I couldn't get it.
No, you should get that icon, play icon.
Yeah, I guess I have to have the original source file here to be able to see it.
Try the double click.
Double click. Right click.
Double click.
Double right click.
Yeah, yes.
Left click, sorry.
Left double.
Left double.
No?
Doesn't work, yeah, already. So what are, so, but what it ends actually is already pretty good. So this is, you can see like a 3D image of the kidneys, and one can see the collecting ducts. You see it here. For example, you see the collecting ducts and then branches, and then you can see it goes the way to the tip. This cyan color, what color is it? So
Cyan.
so the cyan color here, I will come back to that later. So that is a nephron structure. So, but the yellow and then the gray color ones, that is the collecting duct. Oh, actually, it works. Sorry. So actually like the arrow down, so already, so that shows what the kidney, and then so when you scan from the surface all the way deep in, if there's something like Monaco does with the 3D image, then we actually look at this on the embryonic one, right? Yeah, that just shows one individual tip connects to the nephron structure that we will talk later. All right, so this UV branching event, so it can also have problems if you disrupt the branching morphogenesis. One will get a renal hypoplasia. So something one would do, so even in the really young cows, for example, there is also renal hypoplasia from the defective collecting duct development. So for the entire UB, the tip, what it really does, it actually does two things. For now, we talk about one thing is in response to the cap-mesenchyme signal to undergo further branching morphogenesis. That will be a second function we will talk about during nephrongenesis. And then the trunk of directed bud epithelium, so it will differentiate into the collecting ducts. So the tip is, in a sense, is a developmental feature. So it will disappear at the end of kidney development. So in mouse by pitch. So the collecting duct is what's really, the collecting ducts come from the UB trunk. And that is the structure that eventually stays in the adult kidney postnatally. And for that, so its major function is to regulate extracellular volume, osmolality, and then pH. So if one, for example, can have depends, so whether polyuria versus so like when the kidney collecting ducts are not functioning properly in urine concentration. And that is mostly a function of one of the cell types in the collecting duct. So the collecting duct, if you broadly speaking, there are two cell types. That is the principal cells and the integrated cells. Of course, integrated cells can be further divided. So into alpha, beta, non-beta integrated cells. But the principal cells, which are shown here in green, so those cells, those are mainly involved in water reabsorption. So it express aquaporin 2. So aquaporin is a Nobel Prize finding. So integrated cells are labeled here in red. So those cells are mostly involved in pH regulation. So defects in these cells can really generate disease like nephrogenic diabetes insipidus and acidosis. Okay, all right. So then we'll move on to the other urethrative events, nephrogenesis, and then a lot of us actually mostly focus on nephrogenesis. So this.
Do you need to hide them, those things?
Yeah, so that's the already, and then here more. So as we talked about, so the nephrons, this is a repetitive events. So then in humans, so the number we talked about nephron endowments. And then so many of us, like heard last week or the week before, the research day, so the Charlton Labs talks and posters. So, so about nephron endowments. So there is actually a big variation in terms of how many nephrons we sit here. Everybody has a different number. In humans, there can be a tenfold difference. So on average, it's like, people say 1 million, 1.3 million, depends on whose paper you read, all this one, but it can go to from 250,000 to about 3 million. So in humans, that is the variation. In mouse, that's also, some people say like 19,000, I also saw people say 11,000. So all those variations in nephrons, but that is the ballpark. And then, so the nephron number or nephron endowment really so low nephron number can predispose people for CKD, down the road later in life. So the nephrons are formed from the metanephric mesenchyme, which are shown here, which like in this schematic diagram. So UB here, the tip will induce the cap mesenchyme that's capping it. And a subset of this cap mesenchyme will condense. And not shown here, they will first form this tight cluster of cells called pretubular aggregates right under the arm of this newly formed ureteric bud. And then those mesenchymal cell will undergo mesenchymal to epithelial transition. So that's the opposite of tumor formation. So tumor metastasis is you will see EMT. So this is MET. So mesenchymal to epithelial transition, and that forms renal vesicle. And after that, the renal vesicle, which is just a sphere, and it will undergo morphogenesis to form this S-shaped body. And at this point, the distal end of the S-shaped body will also connect with the UB to form a continuous loop. And then, so at this moment, there will be also segmentation. So there's so differentiation of different parts of nephrons. That will eventually, so that will eventually give rise to the entire nephron structure, which I think we are more familiar with here, right? So these are the different parts of the of just showing here one nephron. So this is the renal corpuscle, which actually we normally just when we say it, we say glomerulus. But actually glomerulus is only a part of the renal corpuscle. It's the blood vessel part of the renal corpuscle. But you rarely see people talk about renal corpuscle. We just use glomerulus to refer to renal corpuscle. So the epithelial component of the renal corpuscle actually comes from this proximal most part of the S-shaped body. And then from there, then you give rise, so the proximal tubule will be right next to here, like here in the S-shaped body, it will give rise to the proximal tubule. And then the loop of Henle, and comes back to the distal tubule, which would be here. That's the distal tubule part. If one does molecular doing in situs on the segment markers, one can really see it very well. This the S-shaped bodies, even actually at renal vesicle stage, one can already see proximal versus distal. That axis is already established molecularly.
Here, I will go.
Let's go in, this way already. Okay. So then one would ask, so you have one tip, one cap mesenchyme, and then it gets induced to differentiate into nephron, then how can one generate, on average 1 million nephrons in humans? So that is because there is this, so the capmesenchyme actually contains stem cell-like nephron progenitors. So such that in each round of nephrogenesis, only a subset of them gets induced to the pretubular aggregates. That's what PV stands for. And as I said earlier, it's actually the one that's at the end. So the rest of the capmesenchyme will they self-renew them. So these are this 6-2 genes for us actually use it. To mark the capmesenchyme, the stem cells, the nephron progenitors. So they self-renew, and then they actually also goes with the branching morphogenesis. So it will be always at the most surface part of the kidneys. So that's where we call nephrogenic film. And so with each round of branching morphogenesis, then a subset of them are destined to differentiate, and the rest of them will just undergo self-renewal to generate more capmesenchyme. So that's how one can maintain this rounds and rounds of nephrogenesis. But towards the end of the development, if one look at, for example, the mouse kidneys postnatally, one would see like the capmesenchyme becomes thinner, and then by P4 it will be gone. So after the SG body is so established here, the vasculature will invade. So angiogenesis and vasculogenesis will occur. So podocytes express VEGF, so that induce the vascular invasion here. And then the mesangial cells will also go in, and then so the famous ones, the PDGF receptor beta knockout, so there's no mesangial cells. So, and then the SG body will further differentiate into this stage called the capillary loop stage nephrons, which is also called stage 3 nephron. That's rarely used. So more capillary loop stage nephron is the term that people use. So in this stage, the feature of it is the podocytes are not so so a lot of us are we see a lot of glomeruli, right? The podocytes are all just scattered. And in here, they are more like the baby T cells, and then they are columnar shapes at the outside of the glomerulus. So it's very distinct feature to recognize that's the capillary loop stage nephron. And then after that is the maturing nephrons. So at this stage, the podocytes already form food processes, and they also the cell shape change. They don't look very much like a columnar, like an epithelial cell. So the proximal tubule at this stage also differentiates to show the brush borders for their most distinct function of reabsorption. So one thing I would like to emphasize is that, so we talked about nephrogenesis is a reiterative event. It happens, the induction of nephron happens at the above the tip, also around the tip of the UB. But the most interesting thing is that the kidneys, if you look at a section of a developmental kidneys, it actually has all the structures of the developmental units. So, and then here, so the capmesenchyme is marked here in yellow, and this region that's with capmesenchyme, the UB tips, and there is a third structure, the interstitium. So that's called nephrogenic zone. And then down here, you will see all the developing nephrons. So I think this is about E15 mouse kidney. Such that if you want to study kidney development, then this, you just use one stage of it, you'll see all the structures. So because one can identify, for example, an S-shaped body here, for example, right? And so that looks, that's quite an S here. And then the early nephrons, so one would also see it here and then here. So the deeper down, the more mature nephron structures one will identify. So you just look at an E15 or E16. So some labs use E17 to study it. And you look at it, you identify all these things, the cap mesenchyme, the UB. So every step one can look at which step of development, if you study gene that actually the knockout that gene disrupts. So one doesn't have to look at exactly, even though we initially you will look at it every step, every time point probably, but one can really focus on one time point to identify all the structures and the effects of it from knockouts. So, but that's not the end of the story. So what we talked about, all you see just now in that E15 kidneys, that's pretty much only the cortical structures. So that's because at that point, the renal medulla hasn't started forming. So after that, Lucas would know a lot about so the renal medulla. So after that, after E15.5, there will be a second phase direction of growth. So the collecting ducts and the loop Henle start to elongate towards this pelvic space, and that elongation establishes the renal medulla. So why important of this? This is actually a less studied region of the kidneys, because you see a lot of kidney damages in the cortex, and there's nephron on the glomerular function. But the renal medulla is very important for urine concentration. And so for land animals, like if you compare desert animal versus animals that are like do not have to worry about water intake, so the desert animals will have a much longer renal medulla for urine concentration and therefore water reservation. Okay, so this would be I think it's my last slide. I will just leave this here, and then I think there will be more. No, there won't be any talk about the renal vasculature or anything. Okay, so not in this round. Okay, so then I'll just mention it. So we focused on two major cell types and their derivatives. So the uretic bud, which gave rise to collecting duct, the cap mesenchyme, which gave rise to nephron epithelium. But we touched on the vasculature. So that gave you, so the most important ones for the nephron development, we talked about going into the forms the glomerulus. So in addition to this, there's also a developmentally very prominent cell type is the interstitium. So this population of cells during development is involved in contributes to all these aspects. It contributes to the regulation of nephrogenesis, and they also secrete molecules that regulates branching morphogenesis. And they even plays a role also in the nephron, the collecting duct elongation to form the renal medulla. But this population of cells, the renal interstitium, if one looks at the adult kidneys, it's not prominent. It's you rarely see anything, and once you see it, that's already the myofibroblasts. So that's fibrosis that's happening. But developmentally is a very important population cells. And then another thing is so for example, the renin cell lineage, it actually comes from the interstitial cell population. So the FOXD1, so many of the people working in our lab, you will hear FOXD1, right? So this population of cells, their progenitors express FOXD1. And then that gave rise to the vascular smooth muscle cells, the renin lineage, the mesangial cells. It is actually a very important cell type during development. And then it's more like, so traditionally people think that's almost like a filler. It's just like, filling the space between all these epithelial cells, but developmentally is very critical. And so early in development, so within this interstitium, in the kidneys, we use this word interstitium, there are also vascular cells. So, and that is important for vascular genesis, to establish the vasculature in the kidney. So I think that's all. I want to go, let me see whether I can go back to the title slide. Just, right, oh. How do I go back to the, I wanna, okay, yeah, here,
just-
is it just,
Maybe can I share with the Zoom also?
yes, please. Already,
No, I
Yeah.
can share.
yes.
Is it good?
Yeah, thank you. Yeah, so I come back to this. Does now this slide look familiar, all these stainings? So the red is actually the UB or the future collecting duct. And then the green shows all the nephron image. So you can see the cap mesenchyme here. The S-shaped body, like this one looks very much S-shaped body. And you see the early nephrons, the capillary root stage of nephrons. So the vasculature isn't, and the interstitium are not marked here. So everything here is just the epithelial structures. And then one can see the loop of honey here that's going down into, like lying along the collecting duct in parallel, going down to where this is the pelvic space, the nascent pelvic space, where the collecting duct and the renal medulla will grow into already. So that's what I would like to cover today. Any questions?
I have just a naive question. So you mentioned that there is a great variability between individuals.